Hi, I'm Stephanie Gazowski, Editor of Drug Discovery and Development. Welcome and thank you for joining us for early development screening for technology selection and formulation design. Today's webcast is sponsored by Capsugel and presented by Drug Discovery and Development. A question and answer session will follow our presenter. At any time during the webinar, you can type a question into the panel through the presentation console. We will answer as many questions as time permits. Today you will hear from panelist Michael Grass, PhD. Michael is a principal investigator at Bend Research, a division of Capsugel Dosage Form Solutions, where he develops methods, conducts testing, and analyzes data for a wide range of pharmaceutical formulations. His expertise includes formulation development of challenging compounds, dissolution testing, and physical stability of amorphous dispersions. And now Michael will kick off the presentation.
Thank you for the introduction, Stephanie. So again, I'll be focusing today on early development screening for technology and formulation selection with a particular focus to spray-dried dispersions. So here at Bend Research, where we're part of Capsugel Dosage Form Solutions, which includes several companies across the world with a large breadth of technologies, as you can see here, in bioavailability enhancement, modified and targeted release, as well as other specialty applications. The focus today is going to be in bioavailability enhancement. For us, that includes spray-dried dispersions, hot melt extrusion, lipid-based formulations, as well as several micronization and nanosizing technologies. The recent acquisitions of Excelliance and Powder Size has expanded our CTM manufacturing and micronization and nanomilling capabilities for dosage form solutions. Before I begin today, I just want to acknowledge that several people at Bend Research and Capsugel have contributed to the work being presented today, with lots of collaboration with people around the company. So an overview of the discussion for the talk this morning. We'll begin with a little bit of an overview of amorphous dispersions and spray drying. And then we'll talk specifically about some of the speciation tests and other early screening tests we use for formulation design and technology selection. And then we'll end with 3 case studies outlining some of those tests as they're applied to a few model compounds. So to begin with, as most of you are aware of the BCS classification system, I simply wanted to point out that many of the current drug candidates shown there on the second line of each of these boxes are in what we would call the low solubility compound space. Those to the right, the BCS class 2 and class 4 being low solubility, high permeability, and low solubility, low permeability, account for something like 70 to 90% of current drug candidates in the pipeline, which means that a lot of compounds are requiring bioavailability enhancement or solubility enhancement technologies. Within that space, there are a lot of possible technologies to choose from. In this plot, I've sort of divided it up on the left to sort of physical state properties or particle sizing, things like changing the polymorph of a compound, making an amorphous dispersion, or reducing the particle size by either micronization or by nanomilling. On the right side here, we see a somewhat different approach, which involves solubilizing or dissolving the compound or complexing it with something, things such as using a lipid formulation or formulating with cyclodextrins. And in the middle here are some approaches which lie somewhere in between, such as making a salt form of the drug or making a co-crystal of the compound, for example. And all of these technologies have their own place and work better for certain compounds than others. And there's a lot of understanding of physiochemical properties as well as the target product profile for a compound to understand what kind of technology to choose. Here I'm showing just a very oversimplified version of how we might go about technology selection. So obviously there are many properties that are important to choosing a technology, from the physiochemical properties to the projected dose of the compound to how we want to display that dose to the client, But you see here two critical parameters shown. On the x-axis is the compound log P, or the lipophilicity of the compound, with highly lipophilic compounds being further over to the right. And on the y-axis here is the aqueous solubility, shown here in mg per mil. So at very high solubility, obviously, typically crystalline drug absorbs quite well. As we decrease the solubility, we might need to increase the dissolution rate, but may not need to increase the actual solubility of the compound. And there is where micronization or nanomilling can have a positive impact on bioavailability. As we further drop the aqueous solubility, we have to increase the solubility through some formulation route. And so one of those approaches is to make amorphous dispersions. And you can see there that the compound space over which amorphous dispersions work is quite large. As we move to higher lipophilic compounds, often a lipid formulation can give a better approach to formulating those very lipophilic compounds. So amorphous dispersions have been around for a long time, at least 40 years. They've been known, and there are several compounds on the market. A few of them are shown here at the bottom. And here at Bend Research, we have worked on over 1,000 compounds from preclinical and even pre-animal studies all the way up through phase 3 clinical trials. In the plot on the upper right is a plot I'll come back to a few times, so I'll take a little bit of time to explain it. On the x-axis is the same log P that was shown on the last graph. On the y-axis here is actually a ratio of the melting point to the glass transition temperature in Kelvin. And that tells us something about the driving force for crystallization of a compound. So the higher it is on this chart, typically, with some exceptions, typically these compounds tend to have a higher driving force to crystallize out of both the solid state amorphous dispersion as well as in solution. And so you can see that just based on these two properties, here at Bend Research, all these dots represent compounds we have worked on. We've really worked on a wide space of compounds over our time here at Bend Research. Both high crystallizers as well as highly lipophilic compounds or somewhere in the middle. And I'll go over a little bit in a little bit more detail later on in this plot how we interpret where a compound is on this chart. So the goal of making an amorphous dispersion is to end up with a material that is homogeneous. It's somewhat characterized here by a single TG by DSC, that it's amorphous. There's lots of ways to characterize that. Here is shown powder X-ray diffraction, and having tunable properties. So we want to be able to tune density of the particles as well as loading in the particle of the active, and other particle properties that can affect downstream manufacturing, such as tableting. The process is shown on the left, and really the key focus of spray drying is that we take a solution in solvent and we rapidly dry the particles. And this rapid drying allows us to kinetically trap even thermodynamically immiscible systems into a metastable state that can be useful for a broader range of compounds and polymers than what can be achieved by some other processes such as hot melt extrusion. So as we formulate an SDD, as with any formulation, we have to focus on the performance, the stability, and the manufacturability of the formulation, and we have to do that in phase-appropriate manner from preclinical all the way through commercial launch. Today's talk, I'm simply going to focus on the performance aspect, but of course in a real program we would have to balance performance with stability and manufacturability. So here's this plot again of log P or lipophilicity on the x-axis and this driving force for crystallization on the y-axis. So here I've divided it up into several sections, and the top section highlighted here is the section where we have a high driving force for crystallization, so a high Tm over Tg. And these compounds we tend to formulate at lower loading to try and inhibit crystallization both in the solid state as well as in solution upon dissolution. And a common failure mechanism for these compounds is shown here on the dotted curve, where the concentration of drug increases during a dissolution test or in vivo, and then rapidly precipitates to the crystalline solubility. And of course, the goal here in formulating is to reach something that looks like the pink curve, where we maintain that rapid dissolution rate, but we sustain a high level of drug concentration throughout the course of the dissolution test or throughout the transit in the GI tract. Another section on this graph is shown in the dark green, or over in the highly lipophilic compounds. And here the failure mechanism tends to be a little bit different. The failure mechanism here tends to be slow dissolution rate, particularly as the SDD particles get larger or we increase the drug loading, so we have more lipophilic nature of the SDD. The dissolution rate gets slower and slower. And so here again, the goal of this is to maintain the natural supersaturation of the compound, but increase dissolution rate. And we can either do that by dropping the loading in the SDD, or making smaller particles, or by adding additional components to our formulation that will cause it to dissolve more rapidly. And so that brings me to this slide about how we can formulate spray-dried dispersions. So we have lots of excipients at our fingertips to choose from. The core of making an amorphous dispersion, apart from the drug, is usually a high TG polymer that helps the system be physically stable in the solid state, and then also dissolve rapidly. And so depending on what we want to achieve in our spray-dried dispersion, we can either choose an ionizable compound, and this might be an enteric polymer such as HPMCAS or HPMCP, that have acid functionalization so that it doesn't dissolve in the stomach and only when it transfers to the intestine. Or we might choose a rapidly dissolving neutral polymer like PVP or PVPVA. We might want to select a polymer that has more hydrophobic character, or we might want to choose one that is a good hydrogen bond acceptor or hydrogen bond donor, depending on the compound. Also, in cases where it's needed, we can add a surfactant. A few of them are shown there at the bottom. Those can often impact dissolution rates, but often on the flip side of that, decrease physical stability in the amorphous dispersion. So they have to be balanced with those two needs. So the way an SDD particle dissolves typically takes one of two routes. So the first one I've shown here is where an SDD particle hydrates and phase separates when it gets in contact with media. And so we go from the left on a dry SDD, and it begins to hydrate the outer layers. And then as it hydrates all the way through, we get a phase separation and form these small colloidal or nanoparticles of drug that are phase separated from the bulk polymer phase, and then the polymer rapidly dissolves, creating these tiny nanostructures in solution that can dissolve rapidly. And then depending on the solubility of the drug, these can either rapidly dissolve into solution or can remain in solution for quite some time for very low solubility compounds. This type of mechanism has recently been highlighted in a paper by Paul Harmon and co-workers at Merck in a molecular pharmaceutics paper recently published, and I would recommend reading that as well to understand the dissolution of certain SDD particles. Another thing that can happen if phase separation doesn't occur when the SDD hydrates is that the SDD can actually slowly take on water and remain in a single phase, thus resulting in a slow erosion process similar to, say, an HPMC matrix tablet where the material slowly erodes. So on the next slide, you'll see a video that is actually a 2-hour time-lapse video of an SDD eroding. And so you can watch it over 30 seconds, what happens in 2 hours under a microscope slide when we have an SDD that remains a single phase when it comes into contact with media. And All right, so you probably noticed as well in that video that there were some air bubbles that slowly disappeared, and that's the result of the spray drying process actually creates hollow particles. And then as water comes into that inner pore, that air bubble eventually is released as the SDD particle dissolves. After the SDD dissolves and creates these nanostructures or colloids, then the drug obviously needs to dissolve and then eventually permeate across the epithelial cell wall or cell membrane to get into the bloodstream. And so the species that we consider when we look at SDD dissolution and what we call speciation, which is where is the drug molecules residing in this plot. So we have these colloidal particles shown here down and towards the bottom as these blue spheres. And that's one type of species we consider. Another is the bile salt bound drug that's shown up in the upper part in those little black structures. And then of course, most importantly is the freely dissolved drug shown there in those little gray dots. And is the freely dissolved drug that actually can transport across the epithelial cell membrane. But for a lot of these high log P low solubility compounds, the rate limiting step is often not crossing the cell membrane, but rather crossing the unstirred water layer or the unstirred mucus layer that you can see kind of shown there by that dotted line, where diffusion is rate limiting. And so free drug is the most rapidly diffusing drug across that membrane or across the mucus layer. But drug in micelles can also play a very important role in shuttling drug across the mucus layer to the epithelial membrane. So when we consider different dissolution testing or other tests of our compounds, we also have to consider what we're measuring. And so we may be measuring neutral free drug, which we consider to be kind of the most important for absorption. But we also may be measuring ionized drug, depending on the pK of the compound. Drug in micelles, particularly for high log P compounds. In many of our dissolution tests, we measure drug colloids because they are difficult to separate or to spin out by centrifugation. So they may show up in our dissolution tests or other assays. And then we have, of course, undissolved SDD, precipitate, and maybe crystalline material if our formulation is crystallized. So one of the first critical tests for us is to understand for a compound what is the solubility and how well can we sustain a solubility that's above the crystalline solubility. So in here we have to understand a little bit about what the crystalline solubility is and what the amorphous solubility is. And the amorphous solubility is simply the solubility in solution that's in equilibrium with the amorphous form of the compound when it's saturated in that media. And it's higher than the crystalline solubility because we've broken the lattice energy of the crystal. And so since we have a higher free energy of this form, we can have a higher solubility. And Lynn Taylor has done a lot of work in this area understanding the amorphous solubility over a range of compounds. And this is a plot from her group showing the amorphous solubility of several pharmaceutical compounds ranging from an enhancement ratio. So this is the amorphous solubility divided by the crystalline solubility, or how much enhancement you get by making it amorphous. And you can see here it ranges from 4 to 65 times the crystalline solubility. We have now measured the amorphous solubility of dozens of compounds in our labs as well, and we have seen an even extended range from this. We have seen amorphous enhancement ratios ranging from as low as 1.1 all the way up to greater than 100 times the crystalline solubility. So in our labs, the way we test this is a relatively straightforward test where we dissolve drug in a suitable organic solvent, and then we add that to the media of interest using a syringe pump. And so what is shown in this pink line is simply that we're increasing the total drug added to the media in a linear fashion up to some final concentration, and then holding it constant and looking for possible crystallization or precipitation of the drug. So here on the x-axis is simply the time of the test, and on the y-axis is the added concentration. And then what we do is we look at the UV signal from that's generated in solution, and we see that as we add more and more drug, there's obviously more and more drug dissolved up to a certain point where we begin to see scattering. So here you see that first in the blue curve. And that scattering can be either indicative of phase separation, or what is termed liquid-liquid phase separation, or LLPS, or it can be liquid-glass phase separation for certain amorphous compounds. And we see a scattering event indicative of there being a second phase, or it may actually be crystallization that we're observing. And so careful looking at the test tells us whether we're seeing the amorphous solubility or seeing crystallization. And so here's an example of the data that we get out. So again, it's the same plot of concentration versus time. And so the scattering is shown in red. So if we follow the red plot, we see that there's no scattering for the first 200 seconds or so of the test, corresponding to a concentration of about 170 microgram per mil. And then we see a sudden onset of scattering, and we can extrapolate that back down to reach to tell us what the amorphous solubility is. So that initial scattering event is indicative of phase separation into an amorphous phase, and therefore corresponds to the amorphous solubility. The second, during that time when we're seeing an increase in scattering, the concentration here is also plotted, shown in black, and we see that the concentration is flat during that time. So that's telling us that the actual dissolved drug concentration is not changing even though we're adding more drug, because all of the extra drug is going into this second amorphous liquid or solid phase. And then at some point, we see that the concentration starts to drop and the scattering gets very noisy, and that's indicative of crystallization. So in this particular case, we've seen crystallization after about 80 seconds or so of sustainment at the amorphous solubility. Another test we often run in early feasibility is a dissolution test, but it's an API-sparing dissolution test that we call the Microcentrifuge dissolution test. And so the basic principle of this is that to be API-sparing, we're running a dissolution test in a small Eppendorf tube with 1 to 2 milliliters of media. And we're using mixing and then centrifugation to separate large undissolved species and precipitate from what remains in the media. Because we're using a microcentrifuge for this, and the G-forces are somewhat moderate, we tend to spin down all of the undissolved SDD and precipitate, but we tend not to spin down a lot of the colloidal species that form. So you can see a typical output of this in the lower right-hand corner, where the drug concentration goes up quickly and is maintained. But in this test, without additional sampling or testing, which we often do, such as ultracentrifugation or filtration or other detection, we get these drug colloids, biosalt micelles, and free drug. And that's where additional analyses come in to understand how much of each of these species we have. So a third test I'm going to talk about is a test that is a little bit more specialized, and it's what we call a membrane flux test. And so in this particular case of the test of how we run it, it's similar to a Pampa assay, and we're looking at the drug that comes across to the permeate. But the way it's designed, we've engineered it so that the surface area to volume ratio that we have in this test is conducive to looking at things such as dissolution rate and understanding the importance of colloids to our material. So in this, the flux, or the amount of drug that's crossing the membrane per unit time, is simply the permeability times the concentration in the feedstock solution. And the permeability can be understood as a few resistances in series. So there's the boundary layer resistance in the aqueous feed next to the membrane, and then there's the membrane resistance, the actual diffusion across the membrane, and then we have another boundary layer on the back side of the membrane. And we can engineer this test so that the boundary layer on the backside of the membrane, or in the permeate, is minimal contribution to the overall resistance of drug crossing the membrane. But depending on the compound and how it's run, the feed side boundary layer or the membrane may be limiting. So in cases where the boundary layer is limiting, the critical parameter here is the effective diffusion coefficient across that boundary layer. And so that effective diffusion coefficient is really a weighted sum of all of the drug species that can diffuse across the boundary layer. So this includes free drug, which obviously has the highest diffusion coefficient, drug in micelles, which is a bit lower, and then these colloidal species, which can also diffuse, but they're considerably larger and therefore have a slower diffusion. In cases where the membrane is limiting, there are a few key parameters, but in the in vitro test, one of the key parameters here is how well the drug partitions into the membrane, tending to be better in the cases when we have more highly lipophilic compounds that partition into a lipid membrane better. Another thing we do in early formulation is try to estimate how much drug could be absorbed from an ideal crystalline formulation, such as one that's micronized or nanomilled, and an ideal amorphous drug formulation, where the solubility is higher. So a MAD calculation, or the maximum absorbable dose calculation, is a fairly simple calculation that involves the solubility of the compound and some physiological parameters depending on the species involved, and then the permeability of the compound across the membrane in that species. And that's often, if we have a lot of data and understand that well, that this can be a very powerful tool for understanding whether an amorphous dispersion, for example, can actually solve the problem at hand and reach the necessary dose that's going to be needed to make a product out of the compound. We can also look at things like how many turnovers are going to be needed, and do we need to worry about dissolution rate even for an amorphous form or for a crystalline form. So now I'll go over three short examples here just to kind of highlight some of the use of those tools. So the first compound I'm going to start with is a very low solubility compound, Itraconazole, which is marketed as Sporanox. And so you can see here in the plot in the upper right, again, this compound sits in a space where it's going to be a good fit for SDD technology, but we're very close to the space where we might think that dissolution rate is going to become limiting, as well as close to the space where we think that precipitation may occur as well. And so one of the first things we would go do with this compound is to go measure the amorphous solubility. And so the amorphous solubility of this compound, the test is shown here on the left, and the actual values of it, you can see here on the x-axis in concentration, we begin to see scattering at around 10 microgram per mil. So if we go back to the last plot there, the crystalline solubility in that same media, in FASIF, is less than 0.1 microgram per mil. So the solubility enhancement that we're seeing is greater than 100-fold over the crystalline solubility. As I said, this is an extremely low solubility compound in the crystalline form. So based on the crystalline solubility and the amorphous solubility, we can do a quick maximum absorbable dose, or MAD, calculation over there on the right. And what we see is that we boost the estimated absorption from less than 0.1 grams estimated absorbed in a dog model up to about 4 grams absorbed in a dog model for the amorphous drug. And if we are looking at a 50 milligram dose shown there at the bottom, then our number of turnovers, or our dose number, drops from almost 20,000 down to a little over 100. So we're pretty confident that we'll increase the exposure using an amorphous system here, but possibly not high enough for the intended dose that's being looked at. So the next step here is the dissolution test. And so again, as I said, the amorphous solubility is 10 microgram per mil. And you'll notice here on the y-axis, our drug concentration goes up to 900 microgram per mil, so well above the amorphous solubility. So we know that the drug concentrations we're seeing here are predominantly these colloidal or nanoparticle species. And looking here, we're looking at formulations with HPMCAS with 3 different grades of HPMCAS. So the most hydrophobic grade of HPMCAS is HPMCAS-H, is shown in red, where the concentration comes up to some moderate level of colloids and sustains quite well throughout the duration of this test, going all the way out to 6 hours. You can see, on the other hand, with the more hydrophilic HPMCAS polymers, HPMCAS-M in blue and L in blue and M in green, the concentration of colloids goes up very rapidly, is almost to dose by the first time point, but we see a gradual decrease in the concentration of itraconazole colloids over time. So we see some kind of precipitation or ripening event. These colloids can be further analyzed by techniques like DLS, or shown here, nanoparticle tracking analysis. Using a nanosite, show that the particles are about 130 nanometers. And this is corroborated by SEM, where the particles have simply been dried on an SEM post and looked at after drawing. And they maintain their structure, so they're quite stable. So if we come back to that slide I'd shown before about the different species, then for itraconazole we see that the neutral drug concentration is very low. The ionized drug concentration is extremely low, since the pKa is only about 3 or so. And the drug in micelles is the overwhelming concentration of species outside of colloids, about 10 microgram per mil concentration in colloids, or in micelles. And then the colloids represent most of what we see in the dissolution test and overwhelm all the other species that are formed for this compound. So one thing we want to do is understand the importance of colloids in the HPMCAS-H versus HPMCAS-M and L formulation. And one way to do that, to put that on kind of equal footing, is using that membrane flux experiment. So here are the results of the membrane flux experiment. So again, here on the x-axis is still time. The first y-axis for the solid lines that you see is the total amount of drug that's in the receiver compartment of the flux test. And so here you can see that the amount of drug that's accumulating in the receiver is higher for the HPMCAS-M and L formulations than it is for the H formulation. And then the dotted line corresponds to the right y-axis, which is the flux, or basically the derivative of those solid curves. And what we see is that the flux is about 2 times higher for those more hydrophilic grades of HPMCAS than for HPMCAS-H. And that's telling us that the colloids that are forming are playing a role in this membrane test by shuttling drug across that unstirred water layer, and or by increasing the dissolution rate to provide a higher concentration of free drug at the membrane. So if we come and we revisit our maximum absorbable dose estimation, we see that the membrane flux suggests we get something like a 3 to 4-fold enhancement relative to the amorphous drug alone. And that's by looking at the curves that I showed you, as well as some formulations that form no colloids at all. And we can make an extrapolation about how important these colloids might be. And this is pretty consistent with just simply looking at the equation I had showed before, where we're looking at a weighted contribution of the colloids. So they're in excess of 50 times the concentration of drug in micelles, but because of their lower diffusion coefficient, they don't contribute as much to overall absorption. So for these formulations, we actually were able to dose them in a dog model. All of the formulations that were dosed, we were actually looking at final dosage form effects of HPMCAS-M dispersion. So all of these are actually from HPMCAS-M, just different final dosage forms. And what we see is that for a 50 milligram dose into dogs, we get about 40% absorption, which corresponds to about 20 milligrams being absorbed in a dog model. So this is pretty consistent with our rough estimation that we got from the MAD calculation. and tells us that we're on the right track, and probably these colloids are important to in vivo absorption, although we didn't dose a formulation that doesn't have those colloids. So our second example is ketoconazole. So again, another antifungal, and it has some similarities to itraconazole, being relatively low solubility, though much higher than itraconazole. So the solubility, you can see it down there below. The crystalline solubility is 6 microgram per mil in pH 6.5 buffer, compared to itraconazole, which was in the sort of picogram per mil solubility range. And the amorphous enhancement here, instead of over 100, is a little bit more moderate, being about 30 or 35 times the crystalline solubility. Another critical difference of ketoconazole is that the pKa is much higher than itraconazole. It's at 6.2, so we're going to have a significant fraction of ionized drug in the intestine as well. And you see here that we're in a space where we don't think that precipitation or disarray are likely to be extremely important, although both are always a concern with amorphous dispersions. So one of the first things we went and did here is we looked at a couple different polymers that could be used to form amorphous dispersions. And so here in this amorphous solubility plot, instead of the scattering, I've shown here instead the actual dissolved concentration. So here we see the dissolved concentration on the y-axis plotted versus time. And the two middle curves, the sort of orange and gold curves that say FASF only, are the drug that's been spiked into media without any additional polymer. And we see that we reached an amorphous solubility, and we sustain it for about 20 minutes or so, and then we get rapid precipitation to the crystalline drug concentration. And then we looked at this also by adding 2 different polymers into the media. So the one to the left in sort of yellow and purple is adding a PVPVA to the media. And we see that the induction time for nucleation of crystals actually decreases by almost a factor of 2 when we have PVPVA around. So this polymer is actually inducing nucleation of crystals from the amorphous solubility. On the other hand, when we add HPMC-ASM, the curve shown out to the right in blue and dark blue, we see a delay in nucleation and a slowing of the growth of the crystals shown by the sort of decreased slope of the concentration profile, taking a lot more time to reach the crystalline drug concentration. So if we apply this information now to an estimated human absorption, we see that the amorphous form is likely to be very well absorbed, almost 2 grams absorbed. But we also have to think about the pKa of this drug. It's likely to dissolve very well in low pH gastric environment. Therefore, even crystalline drug of ketoconazole, it may actually have absorption that looks more like this amorphous prediction, because it may all dissolve in the stomach, and as long as we can find a way to keep that from crystallizing in the intestine, we have to get very high exposure. So again, next is data from the microcentrifuge dissolution test. And here is interesting, we're looking at the same 3 formulations that we had for itraconazole. The colors are a little bit different here. So purple is now HPMCS-H, the most hydrophobic one. Red is M, and the green is HPMCS-L. And then in gold is shown 100% pure amorphous ketoconazole. And so you can see here in gray is shown the exposure to the gastric buffer, or pH 2 media, and then transferred into a simulated FASF. And so you can see here that the concentration of HPMC-ASM is apparently much higher than HPMC-SH, and we don't observe any colloids by DLS in this test. So the question is, do we really have 3 times the concentration for the HPMC-ASM formulation, or is it some smaller species that we're not picking up in DLS, or something else that's going on in this test that would suggest that the concentration is not really 3 times higher? So in any case, if we look at the speciation now, it looks quite a bit different from itraconazole. We have quite a bit of neutral dissolved drug, actually, some fraction of ionized drug, and then quite a bit of drug in micelles that can be quite high depending on the dose and exactly what media we're using. So now when we go to that same flux test, we see that the rank ordering of formulations is the same. I've shown only 3 of them here. So we have HPMCS-M with the highest flux, HPMCS-H next, and then 100% amorphous ketoconazole crystallizing out fairly rapidly. One thing we notice here is that the flux of drug from HPMCS-M is not 3 times higher than for HPMCS-H, as we saw for the concentration in the dissolution. And this one reason for this may be that this is not truly all free drug that we're measuring in the dissolution test, and that's why this membrane test can be really valuable for comparing formulations where a dissolution test may be a little bit misleading, and the apparent concentration may not be straightforward to compare between formulations. So again, if we review the predicted MAD, we see that we predict a very high absorption for the amorphous form of the drug, or as I mentioned, as long as we have low gastric pH, even the crystalline drug is likely to match the prediction of the amorphous drug. And if we look through the literature, what we find is that in cases where gastric pH has been controlled and is made sure to be in a sort of typical low fasted state, the maximum absorbable dose is well over 800 milligrams, kind of the highest dose that we can find in the literature. It appears to be 100% absorbed. And there appears to be no precipitation in the intestinal tract there, even for the crystalline drug dissolved into the stomach. However, we do see that the absorption is quite lower for people who have elevated gastric pH, so people who are taking, say, a proton pump inhibitor. And so that's a case where an amorphous form of this compound could actually be very valuable by limiting the effects that an elevated gastric pH would have on absorption, and could be used to blunt that effect and to have high absorption in all populations. So the third example I'm going to look at is erlotinib. And so this one is a bit different because it is much closer to an area where we would say it's a rapid crystallizer. So the log P is a little bit lower than the last 2 compounds, and the TM over TG is a bit higher. So you can see here the PK is about 5.4. So again, it's highly soluble in gastric environment. And at pH 6.5 is relatively low solubility, 1 microgram per mil in buffer and 8 in FASF. And then the amorphous enhancement is about 20-fold. So here again, similar to ketoconazole, we're looking at the amorphous solubility test and looking at concentration versus time plotted here. In black with no added polymer, and in red with added HPMC. And so you can see here that the amorphous solubility is not even reached if we don't add a polymer to solution. We actually crystallize before we reach the amorphous solubility. So this is a very rapid crystallizer. But by adding a little bit of HPMC, we can actually extend the duration of sustainment considerably for this compound. And then for the MAD calculation, we see that in cases where we reach that amorphous solubility, or by dissolving it in the stomach, as long as we don't get precipitation in the intestine, we can reach a very high dose absorbed, much higher than the clinical or commercial dose of 150 milligrams. So in this case, we actually took a little bit of a different approach. Here I'm not showing any SDD formulations, but instead looking at simply what type of capsule we put the erlotinib crystals into. And so here we have a similar test, a pH transfer test, where we start out in pH 2 for the initial part of the test, and then transfer into a simulated intestinal buffer. And in this particular case, in green, I have shown the crystals put into a gelatin capsule, so it's just a standard gelatin capsule. And then in the blue is a VCAPS plus capsule, or an HPMC capsule. And so you can see that dissolution in the stomach is slightly decreased by putting in an HPMC capsule, although it's quite minor. And then upon transfer, we see that we sustain a higher concentration of drug for about 15 minutes and then have much slower precipitation to the crystalline concentration relative to the gelatin capsule. And so this suggests a different way to formulate a highly gastric soluble weak base, wherein we can simply take our sustaining polymer and perhaps put it into a capsule or add it to a tablet as a coating or into the granulation. So here's an approach where we don't actually need to formulate Amorphous per se, as long as we have that gastric solubility to dissolve the drug. So as a quick summary for Erlotinib, again, we have a rapid crystallizer. We want to focus our formulation efforts on a sustaining polymer. And again, we can reach this high degree of absorption by either making sure the drug gets dissolved in the stomach and sustaining that with a sustaining polymer, or we could use an amorphous form. And again, like ketoconazole, by using an amorphous form, we might be able to blunt the effects of a proton and pump inhibitor or other things that may increase gastric pH. So in summary, first of all, thanks for tuning into this webinar. And then also, so on the technical side here, again, amorphous dispersions are a very robust technology for a range of compounds. In early compound formulation, it's important for us to look at all the physiochemical properties of a compound and to use some fairly simple tests to try and rank order possible formulations and understand the likelihood of success for any formulation strategy we might select. The MAD prediction tool that I showed really is kind of a ballpark estimation of what we might be able to expect from different technologies and really understand, again, likelihood of success and predictions that we might see in different animal models. The amorphous solubility test that I showed is really a critical aspect of early formulation before we begin to make formulations to understand what is that amorphous solubility and what polymers we can choose to best help sustain that amorphous solubility for rapid crystallizers. And then the membrane permeation test is really another tool in dissolution testing to further understand speciation and to compare formulations, particularly when assessing the level of free drug in a rapid dissolution test is challenging.
Thank you, Michael, for your presentation, and thank you, viewers, for joining our webcast. Now we have a quick poll question for you to answer. Please take a moment to answer it. Would you like Capsugel to follow up with you after the webinar? Yes or no? We now have a few minutes for our Q&A session, so please take this time to submit a question if you haven't done so already. Our first question is, how is it possible to prevent functional excipients used to control the dissolution profile to negatively impact the overall increase in apparent solubility?
Yes, that's a really good question. And so I think a lot of people know that many functional excipients that increase apparent solubility can negatively impact free drug solubility. So a classic example of this that has been in the literature quite a bit are cyclodextrins, wherein it forms a complex and therefore sometimes can actually lower the free drug or effective solubility, even though the apparent solubility in solution is going up. And of course that depends on the dose and the drug and lots of other things as well. At high dose that won't be a problem. And so some of the functional excipients we might add to SDDs, we may add them in an amount that allows us to increase, for example, dissolution rate, but not actually have any effect on solubility. Or we may add excipients that we want to help increase solubility. Of course, there we have to make sure that we're increasing it in a way that doesn't actually decrease the free drug solubility while it's increasing apparent solubility. So that's a lot of details go into that, and it's very formulation specific. And that's where a lot of the other tests that we also run come into account. We could use a membrane test to understand that, or do some other speciation like ultracentrifugation or filtration to understand what species are actually formed as we add more functional excipients or different functional excipients to a formulation.
Great, thanks, Michael. Our second question, what is the composition of the colloids that form in the itraconazole example?
So the composition of these colloids is predominantly itraconazole. The understanding that we have of them, at least currently, is that most of these drug colloids that form are mostly pure drug, and then they're functionalized by either a polymer or surfactant, depending on the formulation, basically like a surface coating. So we can almost imagine this like an amorphous nanoparticle of drug that has been functionalized by a surfactant or polymer that's coating and therefore keeping it stable.
Okay, our next question. How do you perform amorphous versus crystalline solubility?
Right, so I went over a little bit how that amorphous solubility test works in most of the cases. So in crystalline solubility, what we typically do, and I think it's fairly standard, is we take crystalline drug and we saturate media with crystalline drug so that we have undissolved solids. And we let that stir for some set amount of time, usually a time point at something like an hour in a day or somewhere in there. And then we assay the media with HPLC or some typical method. Here in the amorphous solubility, it really depends on the compound. If the amorphous drug solubility is stable and it doesn't crystallize, there are many ways to address this. You can make amorphous drug and then dissolve it into solution and do the same type of assay you would do for crystalline drug. In this case, we're looking for a robust method that works for both stable and rapid crystallizers. And so in that case, we want to do the test quite rapidly. And so the test I described, here if I can go back to that slide. So in this test that I described, what we're doing is we're actually adding the drug from a stock solvent solution and then looking for when we begin to see scattering. And then we use some other tests to make sure that we're not seeing crystallization. But we're using that as an indication of when we reach the amorphous solubility. So at the onset of scattering, we look at how much drug has been added to solution, and we're defining that as the amorphous solubility. So this is not quite as precise of a measurement as we would be able to do for crystalline solubility, but it does give us a pretty good number on the amorphous solubility, usually within 5 to 10%.
Okay, thanks, Michael. Our next question, you showed an increased sustainment on gastric to intestinal transfer for erlotinib with VCAPS plus. Is this a general phenomenon?
We have looked at 4 or 5 different weak bases for which all supersaturate in the gastric environment or go to a high solubility in gastric environment and therefore are supersaturated in the intestinal environment. And that has been a general phenomenon for all 4 of those compounds that we studied here. There's also literature examples of a compound that is a commercial compound that's dosed as a salt and had higher bioavailability dosed with a VCAPS plus capsule. And some testing in our lab suggests that the VCAPS plus capsule is actually inhibiting precipitation in the gastric environment in that particular case. And then there are some other publications as well looking at other enabling formulations. For example, there's a publication that looks at dosing a supersaturating lipid formulation either in a gelatin capsule or in a VCAPS plus capsule, and that has the same type of trend where the VCAPS plus capsule helps inhibit precipitation both in vitro and increases bioavailability in vivo.
Great, thanks, Michael. Our next question, are the types of tests you described equally useful for predicting the effectiveness of SEDDS SMEDDS, formulations as they are for SDD?
That's a great question. So in our minds, we would say yes to a large extent. The true free drug solubility that's going to be attained with a good SEDS or SMEDS formulation should be the same amorphous solubility that we're testing here. And that being the truly solvated drug. SEDS and SMEDS can change, obviously, the amount and type and number of micelles that form in the gut, and therefore that number will be quite different with those formulations. A membrane test is also very useful for SEDS and SMEDS formulations. One just has to be very careful with what that membrane is. So for our compounds, we showed kind of a typical pamphlet-like membrane. So this is a lipid-filled hydrophobic membrane. That's probably not likely to work with a lot of SEDS and SMEDS formulations that are designed to solubilize lipids into micelles and into colloids that form. And so what tends to be more successful for those formulations in terms of a membrane test is to use an actual biological membrane test, like a CACO2 or an MDCK type membrane assay for those formulations.
Okay, thank you, Michael. We'll need to wrap our webcast, so this will be our last question. You described an amorphous solubility test using drug-dissolved in solvent. How does the solubility compare to the concentration reached with an SDD?
Yeah, that is a good question, and typically, in most cases, I would say 70 to 80% of the cases that we've seen, they match up very well. In cases where they're different, there's usually an underlying reason for that, like slow dissolution rate or other strong drug-polymer interactions that form in the solid state that are difficult to reproduce in that amorphous solubility test where everything is being dosed from solution. But I would say, in general, that they are equivalent or very close.
Okay, thank you, Michael, and thank you again for joining us. This recorded webcast will be available on demand within 24 hours, and we hope you enjoyed the webcast and enjoy the rest of your day.